cells perform a number of metabolic activities. For these metabolic activities, they have certain requirements. These requirements are fulfilled by the environment. In turn, they add a lot of metabolic waste products into their environment. However, to carry out the various functions, the cell should be surviving in an optimal environment because the various variables of the environment like temperature, pH, then the ionic concentration, all these affect the metabolic activities of the cell. So there should be certain mechanisms to maintain a constancy of this environment of the cell. So that constancy of the environment is maintained by homeostasis. See, a unicellular organism is basically in direct contact with its environment, and all its requirements are fulfilled by ingestion from the external environment, and it adds the metabolic waste products into this external environment. But this external environment is huge. It acts as a sink for the metabolic waste products, and it has all the things which are required by these unicellular organisms. On the other hand, a multicellular organism is surrounded by other cells. So you see, this is a mass of cells, right? So the cells are not in direct contact with the external environment, right? So cells are surrounded by other cells, plus you see that in between the cells there is certain space, and here this is the environment, immediate environment of the cell. So whatever products the cell needs, it has to get from this immediate environment, and it has to release the metabolic end products into this particular environment. But if it is so small, don't you think that it will be soon exhausted, right? So there are mechanisms by which this internal environment is kept in a near constant range. The various variables are kept in a near constant range. So you know that there are blood vessels actually going through various organs which actually carry these metabolic waste products and release the contents which are needed by the cell, isn't it? So how this constancy of internal environment of the cell is maintained? So basically, homeostasis is defined as the processes used by living beings for maintenance of near constancy of internal environment of cell. So this is the definition of homeostasis. The processes, right? Then maintenance of near constancy of internal environment of cell. So if you see the above definition, very logically you will get to some questions that what is this internal environment we are talking about, right? Why is there a need to maintain its constancy? And what are the processes which maintain this constancy? In brief, we have dealt these two questions, but we'll clarify a little bit more. But here we will focus more on the processes which are important to maintain the constancy of internal environment of the cell. So this here diagram shows a cell, right? So this is the boundary of the cell, okay? And each cell is actually surrounded by immediate environment, right? Which is basically the interstitial fluid. So it is surrounding the cell. Now, there is a blood vessel which is going through various cells, right? Now you see that the immediate environment of the cell is this, right? And the cell actually takes up nutrients from this. Okay, so nutrients enter here and it releases some metabolic waste products into this environment. Now, this blood vessel basically acts as a bigger environment, right? Because it takes up the metabolic waste products from here and releases the nutrients and other requirements into this interstitial fluid. So basically what we are telling is that the internal environment of cell, or what is known as internal environment of body, is this interstitial fluid, and actually Claude Bernard coined the term milieu interior. for this interstitial fluid, which is the immediate environment which is surrounding the cell. However, now we actually refer to both interstitial fluid and the plasma, which is flowing into the blood vessels, as the internal environment, because as I told you that it is very important that how blood vessels are changing the environment of the interstitial fluid, the variables of the interstitial fluid. So now this extracellular fluid is referred to as the internal environment. Now, as these blood vessels carry the various metabolic waste products and the nutrients, you see various organ systems are helping it to perform its function. Respiratory system is adding in the oxygen and carrying out the carbon dioxide. So ultimately it is being added to the external environment, which we were referring in case of unicellular organism also. Then the digestive system is also bringing in the nutrients, and there is excretion of some waste products, and excretory system again helps in excretion of waste products. The point is that various organ systems are working together to maintain this internal environment, right? So all the systems are actually working for the survival of a single cell, right? Even though each system is formed by cells, right? So it's like many systems are working together. The combination of many cells is working together for the survival of each cell, and each cells, the different cells combine together to form the many systems, right? So it's like many are working for one and one is working for many, right? So for homeostasis, groups of organs perform related functions to basically accomplish a common activity which is essential for the survival of each cell and hence for the whole body, and they are acting together, they are acting along with each other. Plus, this action of different organ system is regulated by endocrine and nervous system in an integrated manner. So fundamentally, you see, cells make up the body systems, the tissues, the organs, right? And these organ systems help maintain the homeostasis, that is the near constancy of internal environment. And this homeostasis is essential for survival of cells. So that's what I was talking, that all the body organ systems working together for survival of each cell, and each cell combining together to form the various body systems. Now, in order to maintain homeostasis, there should be certain systems which should be able to detect deviations, right? So if there is any deviation in the variables of the internal milieu, we should be able to sense it. Then whatever information they have obtained from different areas, they should be able to integrate the obtained information, get some meaning out of it, and then make appropriate adjustments so as to restore these variables to its desired value, that is in a particular normal range, or to a desired value which is suited for that physiological function. So let's take an example of how this is happening. Well, this is brought about by the various components of the control system. So there is a stimulus, right? That is, stimulus is basically the variable. So whatever is the present state of the variable is sensed by a sensor, right? So there can be chemosensor, mechanical sensors, so which can detect deviations in the chemicals, that is the chemosensors, and detect any deviation in say suppose shear stress, right? So that is the mechanosensors. Then this information which is detected by the sensor is conveyed by means of afferents to a control center, right? So afferent actually carries the information to a center. Center processes that information, okay, and then it sends efferents to the effector. So efferent is carrying the information from the center to the effector, and this effector, there are certain changes in the function of this effector so that there is a response. So this response will help in changing this particular stimulus again, right? So let's take an example of maintenance of body temperature. Say suppose there is a change in the body temperature, so that is detected by our sensors, right? That is the thermal sensors are there. which then carry the active information to the hypothalamus, right? Then hypothalamus makes some adjustments, and there may be some change in the effector. So suppose if it is very hot, then it will cause sweating, right? And it will also cause a change in behavior of the person. It will lead to vasodilation also. So that are all the responses which are happening, and the effectors in this case will be the sweat glands and the blood vessels. So these are the components of the control system. There is a sensor which detects the stimulus, afferent, control center, efferent, and effector, which brings about the response. Now, these components of the control system act in various ways, and these ways are feedback mechanisms and feed forward mechanisms. In feedback mechanisms, we have a negative feedback and positive feedback. Let's see these components one by one. So this is an example of feedback control system. So here is the input which is sensed by the sensor. Then the current value of the input or the stimulus is sent by afferents to the controller. Now, controller actually compares it to a reference value or what is known as set point. With this, it makes some adjustments, and via the efferent, the information is going to the effector and there is the output. Now, output is what? Actually, the output is the change in this input only, right? Because of these effectors, there is some response which causes the change in the input only. So the sensors will again sense this output. So because the response is going to change the current value of the variable, isn't it? So the sensors will again sense it. So what is happening? The output is fed back as input into this system. That is why the name feedback, right? Because the output is being fed back as input into the system. So it is not a one-time process which is happening. It is like a continuous process which is happening so that the system is able to monitor the value of the variable continuously. Now, these afferents and efferents can be either neural or hormonal. So these mechanisms, control mechanisms, can be either neural control mechanisms or hormonal mechanisms. And when both these systems are involved, it can be neurohumoral mechanism. So maybe the afferent is the nerve signal which is taking the information to the controller, which in turn is releasing some hormone. Okay, and hormone is causing the various changes, right? So in that case, it will be a neurohormonal mechanism. So feedback control mechanism can be either neural or hormonal. Now, this feedback control mechanism can be either negative feedback or positive feedback, right? In negative feedback, what happens that this control system actually changes the value of the output to opposite side. That means if there is a decrease in the input, say suppose if we take an example of blood pressure, if there is a decrease in the blood pressure, the control system will in turn cause the increase in the output, that is increase in the blood pressure, which will again be sensed back as input, right? So this will keep on happening. So negative feedback system will change the value of the output to the opposite side. And if there is increase in the input, so suppose if the BP has increased more than the set point, then the system will cause a decrease in the output. So that is negative feedback system. So let's take an example what we were talking about blood pressure. So blood pressure is sensed by the baroreceptors, and which by 9th and 10th cranial nerves send the information to the controller, and via the autonomic nervous system, the effectors are the heart and vessels. The information is conveyed to the heart and vessels, which bring about a change in their function, and the BP is changed, and this is again sent back to the baroreceptors. Plus, you see that there may be some external disturbance. That means things are happening internally, but suppose if you change your body position, what will happen? BP will change. Okay, because some external factors will come into play, like gravity will come into play, your muscle contraction is happening, that will come into play, right? So that will also cause a change in BP. So one is the control system causing the change in BP, and the other is the external disturbance which is causing the change in the BP. So both of these will affect the current value of the variable, in this case BP, and that is being detected by the baroreceptors again, right? So that's how this feedback control mechanism tends to correct the variable to nearer to the set point. Now, the mechanism by which this negative feedback operates, there are certain properties which come out. One thing is that when the output is being fed back as input, there is some time lag which is happening, because this entire process is taking some time, right? So suppose somebody has changed the body position. Fine. So say suppose at time 1 they change the body position, and this will take some time. Say suppose the time is 2. Okay. Now this system has changed the value based on this particular input. Fine. But maybe in this particular time duration, the time which has passed from time 2 to time 1, something else has happened. There is some other disturbance which has changed this value, right? So there is basically a time lag. It is not an instant phenomena. Okay, so there is a time lag which is happening in negative feedback system. So by the time this correction has happened, maybe something has already changed this primary value of the stimulus. Second thing is, there is something known as error. The value which this controller gets from this afferent, right? When it is subtracted from the set point or reference value, that is known as error. And it is based on this error value that the controller makes the adjustment. So say suppose, for example, the set point value of mean arterial blood pressure is, say suppose 100 millimeter mercury, but the input which we get is, say suppose 80 millimeter mercury. So in this case the error will be only 20 millimeter mercury. But if there is some phenomena which causes a major change in blood pressure, say suppose this becomes 60 millimeter mercury, the error will be 40 millimeter mercury, right? So in that case, the controller has to make more adjustments. So the response will be more in case the error is more, right? So two things in negative feedback control mechanisms which should remember. One is there is a time lag, and there is something known as error, based on which the controller makes the adjustments. Now, because of the time lag in the system, a negative feedback system actually shows oscillations in the primary variable which is being sensed. So we don't see, like if we are taking blood pressure from moment to moment, it will not be like something has changed the blood pressure. Say suppose there is a rise in the blood pressure and the control system bring it back to normal like this. It's not like this. What happens that if there is a change in blood pressure, the control system will try to correct it, and with correction what will happen that it actually brings it not towards the baseline, but it actually decreases it more because of the time lag, because probably it has sensed this value, but the value is continuously changing, isn't it? So the correction is not perfect, right? Then again this value will be sensed by the baroreceptors. Then again there the control system will act and it will try to correct, but it will again overshoot, right? So this is known as oscillations. This phenomena is also seen in certain disease states in which the time lag actually increases. So one example is Cheyne-Stokes respiration, which occurs when there is increase in the circulation time, right? So this is actually a form of abnormal respiration where person gets hyperventilation and apnea. So the current value of the sensor is not immediately sensed by the chemoreceptors because of the increase in the time lag. So this leads to increase in oscillations in the system. So Cheyne-Stokes respiration is one example where the time lag of a negative feedback control system has increased. Now, there is one other property of a control system that is known as gain of a system, which is a measure of efficiency of the system. That means how well it is actually correcting the error. So to understand it, let's take an example. See, suppose normal blood pressure is 100 millimeter mercury. Okay. And if there is any disturbance, say suppose lot of blood loss occurs, then the BP may fall to 50 millimeter mercury, right? But that is when there is no regulation. That means the control system which we are talking about doesn't exist, right? So that means change without regulation. That means if the system is not there, that is 50 millimeter mercury. We have to just subtract 100 with 50. But in case the system exists, so this is the system existing, the system will try to bring it back towards normal, but because of so much blood loss, maybe it doesn't bring it back toward 100 millimeter mercury, but brings it back only to 80 millimeter mercury, right? So in case of no regulation, how much change was there? 100 minus 50, 50 millimeter mercury was there. But when regulation is there, then also we have some change, that is 20 millimeter mercury. So we have to subtract 80 from 100. So that becomes 20 millimeter mercury. So how much this system has actually corrected? You see, it would have fallen to 50, right? Now it has fallen only to 80, right? So the correction which has been done is 30 millimeter mercury. Okay. So now it is, the error is only 20. So we subtract predicted change with actual change, and what we get is 30 millimeter mercury. Now, gain is defined as correction upon residual error. So it has corrected 30 millimeter mercury, right? That is the correction, that is predicted change minus actual change. It has corrected 30 millimeter mercury, and still the error is 20 millimeter mercury, right? So what could have been 50, now it is 80. So that is correction of 30 millimeter mercury, but is still the error from the baseline. That is from the baseline, error is still 20 millimeter mercury, right? So we have to divide the correction by error, right? So 30 divided by 20. So this gives the gain of the feedback system, that is 1.5, right? So this gain is a measure of efficiency of a system, and different systems have different gain. Now let's come to the next type of feedback, that is the positive feedback. Positive feedback acts a little different from negative feedback. What it does is it changes the value of the output in the same direction as that of the input. That means if input is decreasing, the feedback system acts to further decrease the output, right? So this value is going to decrease further. And if the input is increasing, okay, then again this will, output is going to increase. So some examples of positive feedback system are blood clotting, generation of action potential, parturition, ovulation and lactation. So is this positive feedback system good or bad? See, if it is continuously changing the value of the variable in the same direction, it appears that suppose blood pressure, if we take the same example, if blood pressure is decreasing, then it will continuously decrease the blood pressure and it will not be good for the system, isn't it? However, for certain functions, like in case of blood clotting, where we want the state of the system to change very fast, that means the blood is in fluid state and we want it to become solid very fast, isn't it? So we want that the system should actually go in different direction altogether, and that too very fast. So that in that cases positive feedback works. However, it should also stop after some time, otherwise it will be really disastrous. Anyways, finally let's come to other control system, that is the feed forward control system. In feedback we were talking that the output is being fed back as input. Okay, in feed forward, the disturbance itself is sensed by the sensor. Right, in feedback we were talking, it is the variable value which is being sensed by the sensor. Here, the disturbance which is going to change the value, it has not yet changed the value, it is going to change the value, it is sensed by the sensor, which sends the information to the controller. So the controller can make the changes beforehand itself. So it is preventing the change, the anticipated change in the value of the variable. So that is feed forward control system. So basically it anticipates changes in the regulated variable because it is detecting the disturbance itself. This helps in improving the speed and minimizing fluctuations, because as we saw in the negative feedback system that there are oscillations. Here we are anticipating the change and correcting it fast, right? So this is improving the speed of correction, and this is minimizing the fluctuations in the value of the variable. Now, this feed forward control system is always neural. Remember, like that, right? Because it has to be very quick. Hormonal actions are not that quick, right? So, neural is the feed forward is always neural. And it has one more property, that it can adapt, it can learn actually, right? So, the output which is coming out of the controller to change the value of the variable can change with time. Understanding. So suppose initially if a disturbance is there, and with the disturbance the controller gives some output, but maybe that is not sufficient to change the value of the variable to its baseline value. With the what disturbance is going to change the variable, isn't it? So next time maybe the controller can make more adjustments, so it is learning. So that is why it is known as adaptive control. So, some examples of feed forward control are increase in heart rate before start of exercise, right? See, exercise heart rate is going to change, but this is an anticipatory change, just the thought process of exercise. What happens? It causes change in the heart rate. Similarly, when the food actually enters the gut, what happens? There will be increase in the secretions for the digestion and absorption of the nutrients. But just the smell of food, thought of food actually causes increase in the gastric secretions or other secretions, isn't? So that is also a anticipatory change which is happening. So these are examples of feed forward control. Now, in positive feedback, we said that if positive feedback doesn't stop, then it is going to be disastrous. Similarly, the changes which the feed forward control is making, we don't know that whether those changes are adequate or not. Are they going making the changes in the variable in the right direction, wrong direction? So for that, it is important that the value of the variable is always sensed. Okay. So that is the reason that positive feedback and feed forward mechanisms work under a loop of negative feedback. Okay, so here suppose it is shown a positive feedback. There is a value which is input which is increasing, and positive feedback is further increasing the input. And this positive feedback is continuously going on. However, when a certain critical value of output is reached, this negative feedback starts operating, right? So, positive feedback is working under a loop of negative feedback to keep it in check. Second thing is that positive feedback also stops when there is no stimulus. So, when the stimulus goes off, obviously this positive feedback is going to stop. Okay. Then coming to feed forward mechanism. The feed forward mechanism is making a change in the variable by sensing the disturbance. Now, to see what is the current value of the variable, again you see that this is operating under a negative loop of negative feedback loop, right? So, this again the value of the variable is sensed and fed back into controller. So, this controller is receiving the both the inputs. One is feed forward and by the variable, so that there is a double check on the value of the variable. So in summary, what we saw is there were 5 components of negative feedback system. It monitors the end result of activity continuously, and also it is most common homeostatic system, has time lag which leads to oscillation, then gain determines the efficiency of the system, and this feedback system can be neural, humoral or neurohumoral. Then positive feedback and feed forward, they work under a loop of negative feedback. Then positive feedback is used for switching states quickly. It can be disastrous if it goes uncontrolled. So it has to work under a negative feedback loop, and when the input ends, the positive feedback loop stops. Feed forward control sense disturbance and anticipates changes and makes the changes in the variable. That is like a anticipatory change, and it is very fast, right? And obviously, not to forget that they both work under negative feedback system.